should try practicing on your own before coming to class. So today, if you haven't looked through the lecture slides yet, is basically just practicing doing these sorts of calculations, calculating genetic distances between genes, and then we're going to move in a little bit into creating genetic maps using the map distance that we calculate between genes. Did everybody do this last week? Maybe we should recreate this. So turn to a neighbor quickly. Next 2 minutes, fill in this table again. If you did it Friday, it should be relatively straightforward. If you didn't do it Friday, you might have to spend a little bit of time discussing. So 2 minutes, and then we'll come back together and fill this in. Okay. What questions do you have? Which part was easy, which part was hard? So unlinked, maximum genetic distance. What does unlinked mean? Genes are on separate chromosomes. So that means their minimum and maximum genetic distances are 50 centimorgans. Bah. 50 centimorgans. So if genes are on separate chromosomes, they're going to be 50 centimorgans apart. We talked about this last time briefly. The concept here was that if you have, during meiosis, if you have a tetrad of chromosomes, and you have one recombination event happen, then you're going to have 2 recombinant types and 2 parental type haplotypes, chromatids. So the maximum number of recombinants you get when genes are on the opposite ends of the same chromosome or on separate chromosomes is you get 50 centimorgans. So in the case of unlinked, with genes, say A and B on totally different chromosomes, they will always independently assort, Mendel's second law. When genes are at the opposite ends of the same chromosome, like right here, you always get recombination. So their maximum genetic distance is 50 centimorgans, just like unlinked genes. And what's the minimum? 0. Okay. Questions or clarifications about any of that? All right. The last time we started this little practice exercise, which was just picking random chromatids, drawing crossovers, I went back to my office and actually drew them in a little bit more nicely than I had using my stylus during class. So this was the catalog of all of your suggested crossovers where they would happen on these different chromatids during meiosis. And the question was, given this visual information, what's the genetic distance between these genes? We know now it's either 0 to 50, it's somewhere in that range. So for this set of 8 cells that are going through meiosis, they produce how many gametes? In other words, how many chromatids are up there? 4 times 8 cells. So there will be 32 gametes produced, assuming male meiosis. We're not talking about oogenesis. So there are 32 chromatids. 8 cells go through meiosis, you'll make 32 gametes. Every one of those cells divides into 4. Okay. So we've got 32 gametes. Why is that important for calculating genetic distance? Why do we need the number 32? That's the total. This is the denominator. So calculating genetic distance, recombination fraction is equal to the number of recombinants over the total. So now we know the total number of gametes or haplotypes we're going to look at is 32. So then the question just is, go categorize all of those chromatids as either parental or recombinant. So again, take a couple of minutes, go through this if you haven't done this already, talk to a neighbor, see if you agree, count up how many of those chromatids are parental, how many are recombinant, and then when we come back together in 2 minutes, let's figure out what the tally of the recombinants is. How many recombinant? Go ahead. Ah, good question. So which is the parental type? How do you know which one's parental and which one's recombinant?
It's before they cross over. So it would be capital A, little b, and small a, big B.
Correct.
They haven't recombined.
That's right.
Yes.
So capital A with small b is parental type. So here, there's a parental. Actually, let's just say that everyone that involves a crossing over, for simplicity, just say everyone that involves a crossing over event is a recombinant type. We'll make it easy. Okay. So do that. So in this case, it was the first and the third chromatids that have undergone recombination. So they produced recombinant type gametes. Yep. Question?
Yeah, I was gonna say, just for the parental recombinant, can you just say like if it's 3 and 4 crossing or 1 and 2 crossing, it's not gonna be recombinant? Other than
Correct.
crossing would be recombinant?
So if it's crossing between the pair on the left and the pair on the right, it's going to be recombinant. So that will cause recombination, that will cause recombination, that will cause recombination.
But depending on which ones are you crossing.
Yes,
If
exactly.
you're crossing 3 and 4 or 1 and 2, they're still going to be parental.
So here are some parental types in blue.
Yeah.
Okay. So the reason these on the bottom are parental type is because do they generate chromatids that have a different genotype than originally existed? No, because we started here with a little a coupled to a capital B, and with that sort of recombination event, you wind up with a lowercase a coupled to a capital B.
Would these two be different? The third one on each row? Because it seems like even after recombination, they still have the same genes.
They have the same genes, but they have different combinations of those genes. So in this top case and the bottom case, you have a creation of a new haplotype, little a with little b, and capital A with a capital B, which weren't haplotypes that were initially in that cell.
But you're gonna get the same effect on the bottom one?
That's correct.
Okay.
The bottom one's exactly the same situation. Yep. And this is the reason that I'm glad that all of you suggested these different chromatids to recombine is because we did get some that were the same as here. That's going to happen by chance during meiosis because millions of cells in our bodies, well, some of our bodies and not others, undergo meiosis. So by chance, of course, some of the cells are going to have exactly the same recombination patterns. So how many different recombinant types do we have? 1, 2, 3, 4, 5, 6. What else? There's recombination here. 7, 8.
I think the top left and then the second to the right are the same too. I mean, second on bottom.
11, 12, 13, 14. So are all those ones I circled in blue recombinant chromosomes, or did I mess up at all?
Do you want just all the chromosomes total or like the different?
I want the number of recombinants. Sir,
I just had a question.
please.
With the percentage that we get out of this one,
Yep.
how many trials, like how do you, what do you guys go to like find the exact mapping? Because on this one we had two of them that were recombinations between sister chromatids, but how common is that versus a recombination and other ones, like going for all of that?
So the important thing here to understand about how you actually do genetic mapping is sometimes by chance you do get these recombination events that happen between the same chromatids, the sister chromatids. And that part of that is taken into account in the genetic calculation of the genetic distance. So you don't have to worry about it. We wind up calculating a percent recombinant recombination fraction, and that's what we report.
So there's a total of 12 recombinants?
I got 12 too initially, and so that's why I asked, did I screw up somehow when I circled those 14 different chromatids?
Well, the first one on the left side, you get two parental.
In the bottom left corner, those are the same ones.
Ah,
I think they were circled for a different reason though.
blah. Good. 12. So the ones that have circled in blue now are the 12 recombinant types. Now, I want you to realize that the only reason that I'm showing you this is because I can test you on your concept and understanding of recombination by showing you crossing over in meiosis. But practicing geneticists don't ever see this. We don't cut out people's gonads and stare at their cells as they go through meiosis. What do geneticists, oh, sorry, comment on that, yes.
I have a question about the
No,
numbers. So you
that's
said-
fine.
So you said there's 12 recombinants?
Yeah. 5, 6, 7, 8, 9, 10, 11, 12. Yeah. All the ones that I circled at the bottom of the chromosome in blue, those are chromatids that are taking part in recombination that's producing new haplotypes of chromatids. All the rest are going to be parental type. So what did geneticists do instead of opening up gonads and looking at crossing over patterns on chromosomes? Which you can do. You can actually use microscopy to look at this, like is drawn up here. What do we do instead?
Can you show how you got 12 again?
Yep.
Look at phenotypes.
We look at phenotypes. So a geneticist, and that's what we're going to practice next, is instead of knowing genotypes, all we look at is the phenotypes of the offspring, and we then infer, as we've done in the past already, we're going to practice again some more, we infer, we make educated guesses about the genotypes. Okay. So if we number these 1 through 8, for easy reference, somebody tell me one of these cells that's undergoing recombination that doesn't create new haplotypes. So I heard number 5. So when those two chromatids undergo crossing over, that creates physical linkages between the chromosomes like that. So we have a little a connected to a capital B, like there. And on the other sister chromatid, we've got now a capital A connected to capital B. So no haplotypes have changed. We started with a little a, big B pair of sister chromatids, and now we're where we started. So no recombination. Recombination is occurring, crossing over is occurring, but we're not making new combinations of alleles by doing so. Which must mean there's another reason for cells doing crossing over, which there is. It has to do with the segregation of chromatids during meiosis, to make sure that those 4 chromatids stay together and then separate appropriately to produce gametes. You'll get more of that when you do biology 103, cell biology. So I'm not going over the real reason for crossing over. Geneticists would say the real reason for crossing over is to make new haplotypes, to make genetic diversity. But the cell biologist might tell you, well, it's because if you don't do crossing over during meiosis, then the chromatids might not segregate properly into the different cells, the different gametes. So there are really 2 main functions for crossing over. Depends on who you ask. So if we have 12 out of 32 recombination fraction, what is that when you do actual division? What's 12 divided by 32? Pardon? 0.38. So recombination fraction of 0.38, which is 38 centimorgans. Now, if I had just shown you this picture without all the crossing over locations and events, what should you have guessed that their genetic distance would be? Yeah, 0.5 or 50. Genes at the opposite ends of the chromosomes, on average, should be 50 centimorgans apart. Why do I make the emphasis on average? Well, it's most common, they're closely linked.
Smaller chromosomes, even if they're opposite genes.
Ooh, interesting question, or interesting point. Does the size of the chromosome affect recombination? Bigger chromosomes, smaller chromosomes? What's the max? It doesn't matter. Maximum distance between 2 genes, because every meiosis, every set of chromatids will undergo 1 recombination. So even if it's a really short chromosome and the genes are physically close together, there will still be a recombination event between them every meiosis. So short chromosome, long chromosome, that's why we make genetic maps, is because genetic maps use a different measure of distance than base pairs between genes. We look at amount of recombination between genes. So the on average part I'm emphasizing because if you only look at 8 meioses, this is not giving us necessarily a representative view of where crossing overs are going to occur and which chromatids they're going to involve. So if we looked at an infinite number of these cells, then we would wind up getting to 50 centimorgans. We just haven't taken a big enough sample yet to see that. So that's one way to calculate genetic distance based on visual representations of recombination. Okay, so I just made the point a couple of minutes ago that back in the day, and still today, depending on the organism you work with, we don't necessarily have genetic information about the organism. So if we want to learn something about genetic distance between genes, whether they're on the same chromosome or different chromosomes, how do you figure this out? It's all about phenotype. So what would be the let's start at the top and work our way down. We don't know anything that's in parentheses. If we're Mendel, for example, or Punnett or Bateson or any of the other now ancient geneticists. We don't have these genotypes. We don't have P0 genotypes. We don't have their gamete haplotypes. We don't have the F1 genotypes or haplotypes. All we know are the phenotypes. So what were the phenotypes that this cross started with? Capital P is dominant, produces purple flowers. And what was lowercase l? That was something about the pollen shape. Yeah, it was short or round. Let's call it short because L is long, but it might have been round too. So that's a purple flowered short pollen pea plant crossed to a red flowered long pollen grained plant. And how do we know that those are homozygous genotypes here?
You get the same.
Yeah. How do you figure out whether or not a genotype is homozygous? Especially if you're working with a plant. Yeah, plant its seeds. So if you self the plant and you find out if it's pure breeding, if all of its offspring look exactly the same, then you know that it's got a homozygous genotype. So it turns out that that's what, again, we've talked about this before, that's what Mendel used in his crosses, were pure breeding purple short plants and red long pollen plants. So even though he didn't know anything about DNA, what it looked like, what it was, he knew at least that those plants bred true. There was no variation within that plant. Every time it had offspring, its offspring looked exactly the same as the parent. Okay, now we can figure out the haplotypes of those gametes, but only with the benefit of knowing about DNA. So that's something we have to infer based on the phenotype. If you've got a pure breeding, purple flowered, short pollen plant, you can go from that phenotype to the genotype, and then from the genotype to the haplotype of its gametes. This is what you do to set up Punnett squares, so this is all review. What do the F1 plants look like? What are their phenotypes? Those dihybrids. They've got a capital P, they've got a capital L, those are the dominant alleles. So all of the F1 produced from that cross are going to be purple flowered long pollen. Who do we cross F1s with to do genetic mapping? The tester, which looks like what? Tell me what the plant looks like. Here's its genotype, little p over little p, little l over little l. Red and short. Red flowers, short pollen grains. All right. And a lot of you figured out how this works already. Ooh, wow. F2 phenotypes. So when Mendel set up this cross, or actually when Bateson and Punnett set up this cross, after Mendel, remember, you don't need to memorize names. What did the plants look like? What were the F2 generation phenotypes that you get out of that cross? What did the plants look like?
Half are purple long, half are red short. Or no, there's actually a percentage.
So if you've got a dihybrid genotype, what are the 4 different haplotypes that are produced? Capital P with capital L. Capital P with little l. Lowercase p, capital L, lowercase p, lowercase l. Right, those are the 4 different gametes that get produced by a dihybrid. Review. Right, setting up a Punnett square, essentially. So what do those plants look like? Given those haplotypes, we know that the other parent, this tester over here, contributes a little p and a little l. To every genotype. So now hopefully you can tell me what those F2 plants look like. What's the top one? What's the top plant look like? Purple and long, because it's got a capital P, it's dominant, so any plant that has a capital P will make purple flowers. And the capital L gives it long pollen grains. So you figure out the other three. We won't do that at the moment. So those are going to be the F2 phenotypes. What those F2 plants look like, you can put in that column over there. And I've already written out the genotypes. Right there.
Can you always assume that the first letter comes from the first, like, can you always assume that notation the way that we saw?
We don't have to do it this way. That's a good question. So can you always assume that the first letter comes from a certain parent?
Yeah.
No. So here's what you might see. So instead of putting the capital P first, we could put that lowercase p first and a capital P next to it. Okay. Does it matter? Because when we want to figure out, the goal is to figure out from this F1 individual, from this dihybrid, right there, the way we look at its gametes and what they're doing, whether or not they're crossing over, is not like what we did in the previous exercise where we looked at the tetrads and actually saw where crossing over was occurring. What we need to figure out, the haplotypes of the F1's gametes, is to look at its kids and figure out what phenotypes they are, because that gives us all of the information about what alleles were being carried in the F1 dihybrid's gametes. So what do? We know that the tester, all of its gametes have the little p and the little l. Because we know when you self it, it produces all individuals that look exactly the same as itself. And it's the recessive phenotypes. Hence, it's a tester individual. And that's why when you get the F2, one parent's the tester, one parent's the dihybrid, we know what alleles came from the tester. It was a little l. Right? So we know that little l, one little l in every F2 didn't come from the dihybrid parent, it came from the tester parent. Likewise, what about the P? One, and only one, small p, came from the tester parent. So regardless of which order, big P or little p, big P on the left, big P on the right, it doesn't matter. In the genotype, you take one of the little p's and say that one came from the tester. Right? Tester always gives one lowercase allele at every locus you're looking at, every gene. So what's left here, big P with big L, big P with little p with big L, little p with little l, are the haplotypes or the gametes that were produced by this F1 individual. Those are its gametes, the haplotypes. We kind of figure it out backwards. Look at the phenotypes, we know the genotype of one of the parents, the tester, and we can use that to figure out what was inside the gametes that were being fertilized by the tester gametes. What's the last thing I would need to tell you, and then I'll get your question. What's the last thing I would need to tell you to calculate genetic distance?
The parents.
So there are two more steps after this point to calculating genetic distance.
Oh, which ones are recombinant?
Exactly, both right on. We need to know which of these are parental and which of them are recombinant, which of the four. And we do that by comparing those F1 haplotypes to who? P0 haplotypes. So we got the P0 haplotypes up here. And we've got the F1 haplotypes here. So big P with little l is parental. And little p with big L is parental. The other two are recombinant types. And then the other thing you need to know, as you said, was you need to know how many of those 4 different phenotypes of individuals, purple long, purple short, red long, red short, did you actually see? Then you do recombinance over total. So,
Yes, I've realized in some of the exercises, you cross F1 always with a tester, right?
right.
Is that always gonna be the case? Because in that case, it's easy to know the other organism haplotype. But for example, what if you give me like a dihybrid or something instead of a tester? Because I'm kind of
You
afraid
cannot,
that's
yes,
gonna
you
happen.
should be afraid because, oh no, you should not be afraid that that's going to happen
Okay.
because geneticists don't do that because we don't have a way of figuring out, you can't cross 2 dihybrids together to do genetic mapping.
Yeah, that's what I was thinking.
Because then you've got 2 variables. You don't know which
one?
the haplotype of either parent's gametes.
Okay.
That's why we use the tester. Always use a tester. Because we know the haplotypes of its gametes with certainty, for sure, with certainty. We know that 100% of their gametes are going to be little p, little l in this case. So it'll always be a something hybrid crossed to a tester. Monohybrid, dihybrid, or as we're going to see for next class, trihybrid. Whether you like it or not.
Can you repeat what the 2 things we needed to finish your problem were?
Yeah, so at this point, the 2 pieces of information you need to know, one of which we already know because we have the haplotypes of the P zeros. You need to know which haplotypes are parental types and which are recombinant. And you get that by comparing the F1 haplotypes to the P0 haplotypes. And then we actually would need to know, I'd have to tell you something like you saw 50 purple long plants and 25 purple flowered short pollen plants and 50 round flowered, red flowered long pollen plants and 30 of the bottom. And then with those numbers, you can actually calculate a genetic distance. Since I just made them up, that might not actually come out well, so I don't suggest trying it necessarily. But you can and see what sort of a genetic distance you calculate. So we don't need genetic information to do genetic, we just need phenotype information. Now, a while ago, I asked this question. So at this point, hopefully everybody's at least comfortable with the concept. We'll still do some more practice, but the concept of how to do genetic mapping. Number of recombinants over total. And then we can draw representations of chromosomes, except we don't call them chromosomes, we call them linkage maps. Or genetic maps. These are not the same as physical maps. These are representations of the locations of genes based on how much crossing over occurs between them. So we said that the distance between genes A and B in the video that you watched for simple genetic mapping was 40 centimorgans. Okay. So where do you put gene C if the distance between it and B is 10 centimorgans? To the right? Okay, so we could put C over there. That would be 10 centimorgans from B. That works? Yeah?
Can we also do it slightly to the left?
Sure. Be creative. C could also be in between A and B, right? That's consistent with the data we have so far. A to B is 40 units. We could say B to C is 10 units. Okay? We've gotten nowhere. We still don't know the actual relative order of A, B, and C. What information do we need to figure out the distance from A to C? We need to do another cross. So So we looked at one cross where we crossed a dihybrid that was for genes A and B. The second cross to get the B to C distance was a dihybrid containing genes B and C. So we need a third cross to be able to figure out where is C. Is it in between A and B or is it on the right? Remember, genetic distances are additive. What would you predict that the distance between A to C is depending on where it's located, in between A and B or on the right of C? If C is to the right of B, what's the distance from A to C? 40 plus 10, 50. If C is in between A and B, it's going to be 30. 40 minus 10. So we should be able to do a cross and expect we're either going to find the A to C genetic distance is either 30 or 50. And that's going to tell us where to draw C on this map. Any questions about that? So that's the next step, is to do a third cross. Which we'll get to. We'll figure out the genetic distance from A to C. And spoiler alert, for next class, we look at a 3-point cross, which is the video to watch for next time, and we'll do some more examples in class. Instead of doing 3 separate crosses, a cross with A and B, and a cross with A and C, and a cross with B and C, there's a way to do all of this information, collect all of this information in a single cross with a trihybrid. And that's what the video for next class is all about. All right. So I know some of you had some questions about these questions that I left at the end of last class's PowerPoint slides. So does anybody want to raise any of those questions at this point? I'll tell you the answers at some point, but did somebody get stuck working these practice questions, want any feedback? Well, take half a minute and answer the question that's asked. Okay, discuss with a neighbor. We'll come back together. I think we've already talked about this today, actually, so it should be a pretty quick discussion. Okay. Share with the neighbor, half a minute. Okay, so what is it we need to know? I've heard the right answers floating around. So if you're going to answer this question, I've already given you one of the pieces of information you need. Numbers of offspring, different phenotypes. So we need to know something about dominance and parental or recombinant. So what information could I give you that would convey that information? What would you do to figure out which are recombinant and which are parental haplotypes?
You need to work backwards
We need to work backwards from
the F2 generation.
So what is the F1? Let's come up with some letter designations for these alleles controlling flower color and stem length. For B for blue, okay, so B for blue. Okay. So we're going to say, well, we need to know so let's say blue is dominant to red. So we're going to call that big B is dominant to little b. So I would tell you that. Or you would have to do some sort of a cross to figure out dominance.
Based on phenotypes, wouldn't you be able to tell?
Sometimes you can tell based on the phenotype ratios, but sometimes you can't. Which one's dominant and which one's not. So let's say that's true. I'd have to tell you that again, or you'd have to set up a cross. Let's do long is dominant to short. So long stems, capital L, is dominant to lowercase l, short stems. Okay, so there you go. I've given you information about dominance. What else did you want to know? Parental versus recombinant type. So let's look at that F1. It says F1 dihybrid test cross, the bottom line there. F2 phenotypes resulting from an F1 dihybrid test cross. So what's that? Draw out that cross. What are the genotypes involved? It's a dihybrid. So a capital B, lowercase b, dot. We don't know if these are linked or unlinked. That's what we're trying to figure out. Capital L, lowercase l. Cross 2. That's the dihybrid. Always, F1.
Purebred recessive.
Yeah, purebred recessive. Dihybrid, and then the tester individual. Lowercase b's, lowercase l's. What would its phenotype be, by the way? The tester. Red and short. Okay. So we always use the tester individual. That tester individual we order from a greenhouse, maybe. I don't know. So the question that you need to know the answer to get recombinant versus parental type is what sort of a cross can you use to set up to make that dihybrid? Why is that an important question? What's the answer? So what cross was used? We need to know the gametes that fuse together to produce that individual. So what are the genotypes of the parents?
You don't know? Pure breeding.
Dominant and recessive.
Exactly. So if I summarize everything I just heard, I heard it depends. No, that's not summarizing. Has to be pure breeding, but we don't know. This should remind you a lot of a couple of questions that were on the test, asking you multiple ways to set up a cross that results in a dihybrid or a trihybrid. So I'd have to tell you what cross. So it could be capital B over capital B. Let's see. I'm just trying to stick with my notes here. Capital L over capital L. So if that's the genotype of one parent, what's the genotype of the other parent? Homozygous recessive. Would create a dihybrid. Okay. But you don't, the point is you don't know the genotypes of the parents just because you've got a dihybrid. Could be, there are a number of combinations.
Kind of with that, if we're looking at just one individual, can you have two heterozygous parents and that's just one of the offspring, or are we saying that that is the only?
That is the only, has to be the only genotype of the offspring. Has to be. Otherwise, genetic mapping doesn't work. So we have to have those pure breeding P0s that produce 100% of their offspring are the dihybrid genotype. And then we have to cross those dihybrids with known testers to get F2s. So with that information now, you should be able to calculate genetic distance between P and L, B and L. Yep.
You said that the parents have to be pure breeding, right?
Yes.
But as you said on the test, it could also be capital B's and little L's.
Yep.
Times
That's pure
little-
breeding.
That's two pure breeding.
Yep.
So then we could also use those types to figure out the dihybrid.
Right.
There-
So there are a number of different crosses. The only requirement for these P0s is that they be homozygous at each locus. And that when you combine the two parent haplotypes, you produce a dihybrid genotype. Now I want to save a little bit of time. I know I'm at the end of class. I want to show you really quickly the results from the exam, the grade distributions. Then we can get out of here. But now you've got enough information to do this practice question and the other few that are in the PowerPoint slides. So that's what I would suggest doing for next time. Work through these. All the information you need is given. Okay. For those of you that are interested, there's the grade distribution. So it's better than last time. Almost got students with 100%, not quite. But this looks a little bit more like one would expect out of a set of questions. Not quite sure how I would curve this yet. I'll tell you at some subsequent point in the future. But if you're in the top 2/3, you should be feeling happy. So A, B, C maybe goes down to 6 or 5. If you're 4, 3, 2, let's talk. Okay? I've got office hours now for an hour, and then I'll see you on Wednesday. One video to watch for next class.